My name is Jeanne F. I'm a professor at the Scripps Research Institute, and I study stem cells. What I'm going to tell you today is a set of tools that we've developed to try to make stem cell therapies as safe as possible. Let me show you. My first slide shows what my lab's vision is. We plan to use genomic tools to drive discovery and translation of pluripotent stem cell-based therapies. And that Venn diagram you see is our overlap between pluripotent stem cells represented by that tree and genomics. The first on that list in the overlap is autologous cell therapy for Parkinson's disease. And that's what I'm going to focus on today, because this is a therapy that we plan to take to the clinic relatively soon, and so a lot of our effort has been focused on developing tools to make sure that the therapy is safe and effective. But we are also working on drug development using a multiple sclerosis model. We're trying to understand the basis of Fragile X and autism by using iPS cells from people with Fragile X syndrome. And with the San Diego Zoo, I am studying methods for using iPS cells to generate gametes so that we can try to rescue the northern white rhino, which is a species that is going to be extinct in our lifetime. So you probably already know, but I'll remind you that the sources of pluripotent stem cells are embryonic stem cells, which were first made from humans in 1998. That is a picture of or drawing of an embryo. The cells that are shown by the arrow, the inner cell mast cells, are placed into culture and expanded, and they give rise to human embryonic stem cells. In 2007, Shinya Yamanaka developed a method for making the same cell type, but making it from individuals from their skin, and it can also be made from other cell types in a human body, which means that we can make personalized induced pluripotent stem cells from every individual. That involves, in our lab, in making dermal fibroblasts first, and then using Yamanaka's reprogramming factors to turn the cells into pluripotent stem cells. And this is my diagram again of showing the tree to represent all the things that pluripotent stem cells can give rise to in the body. And up in the upper right-hand corner, you'll see that one of those cell types is dopamine neurons. The reason that, that dopamine neurons are important is because they're involved in a regenerative therapy for Parkinson's disease that we've been developing for about the last 6 years. So Parkinson's disease is caused by the loss of a particular type of neuron in the brain, a dopamine neuron in a particular part of the brain, which is the substantia nigra. This little tiny bit of the brain makes all the dopamine neurons that project, that is, send axons into the striatum, which is one of the nodes in this network that leads to control over fine movements. I'm sure you know that the symptoms of Parkinson's disease are loss of fine motor control, which is manifest either by tremors or by a freezing and being unable to move, or both, alternatively. There are a lot of people with Parkinson's disease. It's the second most common neurological disorder. 50,000 or more Americans are diagnosed every year, and they aren't diagnosed until half of their dopamine neurons are already gone. It is a progressive degeneration of these cells, and so by the time they're diagnosed, they're already halfway through their supply of dopamine neurons, and they will continue to die because the drug therapy, which is L-DOPA, does not stop the loss of neurons. The drug therapy has been around since the 1960s. L-DOPA revs up the remaining dopamine neurons so that they can function better, but it doesn't stop their loss. So currently there's no disease-modifying treatment at all. So we propose that dopamine neuron replacement may be able to give the patients decades-long therapeutic benefit. Now this is just a slide, this is a figure from a paper that was published last year by Roger Barker and colleagues about the 4 groups who are sharing plans for our stem cell-derived dopamine neuron replacement for Parkinson's disease. I'll just, you don't need to read all the stuff on there, but the important things are that Roger Barker and Malin Parmar, who are in the UK and Sweden, are using human embryonic stem cells. And since they will not match the patient, they will need to use immunosuppression for at least 1 year. Lorenz Studer in New York is also using human embryonic stem cells and will require immunosuppression. Jun Takahashi, who's in Japan, is using a single induced pluripotent stem cell line, but it won't be a perfect match for the patients, and so he will also be using immunosuppression. And lastly, my group here in La Jolla is using autologous iPS cells, that is patient-specific iPS cells. We're making the cells from each patient so we can avoid immunosuppression. This group is called G-Force PD, and it meets every year or so to coordinate their efforts to take these therapies to the clinic. So this is an overview of our very simple approach. The upper left-hand corner shows the cover of Stem Cells in Development, and that journal has in it the paper which is cited at the bottom, which is an overview I wrote of and rationale for why we're using autologous pluripotent stem cells. We call this a neuron replacement, not a stem cell replacement. It's important because that differentiates us from all the clinics that are working without FDA approval using stem cells that they get from people's own bodies. So there's the brain again on the side, and then on the right-hand side, it just shows the steps. We make induced pluripotent stem cells from each patient. We use those iPS cells to generate dopamine neurons, and then we transplant them back to the patient. The site of the transplantation will be the striatum, which is the place where the dopamine neurons in the substantia nigra make their synapses. The reason we chose autologous neuron replacement was that it had certain advantages. Chief among those is that no immune suppression is needed. This will decrease the problems that the patients have from infections that are caused by the immunosuppression or that are allowed to develop because of immunosuppression. And it also, because of that, will lead to lower care costs for the patients after they receive the transplant. Also, there's no scale-up required. We only need 2 to 5 million neurons for each transplant. That means that unlike CAR T therapies or other therapies that are meant to replace certain cell types, like cardiac cells, for example, we don't need to make 1 billion cells. That will make our manufacturing costs lower, and it also makes it easier for us to do quality control. And importantly, no rejection will happen if we have to redose the patients. So we don't know exactly how many neurons are going to be required. We plan to transplant sufficient neurons so that they will alleviate the symptoms of Parkinson's disease. But if we discover in our phase 1 trials that people are not fully recovering, we can, in fact, re-inject dopamine neurons because they will not be rejected. They're self, not non-self. And also, since we bank the iPS cells from each patient, in theory, those same cells could be used to make other cell types for other cell therapies in the future. There are, however, some challenges. We need to get the iPS cells made from every patient. We need reproducible methods for dopamine neuron differentiation. In other words, we need methods that work for every cell line, not for a single cell line. If we needed to use quality control using animal studies, it would be too expensive. So we need to have quality control methods that can be substituted for each cell line being studied in animals. And importantly, we will need to look at multiple cell lines, all of the cell lines, to find out whether they have potentially dangerous mutations. This next slide is an overview of our quality control, which is what we've mostly been working on besides our differentiation method. We've developed, now this is where the genomics comes in. I have 3, across the top is the method from iPS cells to an intermediate stage. We're telling the cells now that they're going to be neurons and that they need to be a particular type of neuron that is in the bottom of the early embryo's floor plate, the bottom of the neural tube. And then we tell the neurons that they're supposed to make the exact type of dopamine neuron, which eventually will give rise to the substantia nigra during fetal development. This takes 25 days, 18 to 25 days, and this is the stage at which we plan to do the transplantation. At this stage, the neurons have little tiny processes, they're not mature, and so we get very good survival of the cells. They're not all dopamine neurons yet, but they're also no longer dividing. So it's a sweet spot in this differentiation scheme to get the maximum surviving neurons for transplant. But we do keep the cells longer because we want to make sure that the cells that we have in those culture dishes are capable of making mature dopamine neurons. So you'll see those little triangles underneath. The blue is conventional quality control, which uses markers like immunocytochemistry, which is actually shown in that upper path, flow cytometry and RT-PCR. And this is to make sure that the cells pass the criteria to take them forward, whether it's taking them forward through the differentiation scheme or taking them forward to transplant to patients. The orange triangles indicate where we have introduced genomic quality control. The genomic quality control, which I will talk more about in a second, is gene expression profiling and whole genome sequencing, which we feel is important because we do not want to put any dangerous mutations into people if those cells are to last for the rest of their lives, which is what our plans are. We make sure that the cells are dopamine neurons by the green triangle there, the mature neurons. We confirm that they are dopamine neurons by electrophysiology, that is the electrical activity of the cells, which is typical of dopamine neurons of that specific type, and also by making them release neurotransmitter and making sure that they release dopamine and not other neurotransmitters. So essentially that's an in vitro functional test. Our genomic quality control is done at 4 stages. As you see in that circle, reprogramming is the first stage. Second stage is quality control of the iPS cells. Number 3 is the expansion of the cells, and I'll tell you in advance this is the most dangerous part of the entire process because this is the time at which mutations occur in the population. The next stage is differentiation, and then at that stage we want to make sure that the cells are what we hope that they are, that we can we can understand their identity and check one more time to see whether they have any mutations. This is all done before the cells are transplanted. At this stage, we freeze the cells down so we can do all these quality controls before the transplantation of the cells to the people. I'm just going to go briefly through the methods that we've developed for reprogramming. The chief concern about reprogramming was that reprogramming cells might introduce mutations. We showed that with this paper, which we published in 2016, that it is unlikely that reprogramming will introduce any dangerous mutations. You do see introduction of variants for reasons that you will see later. But the important take-home lesson is that reprogramming is benign. It doesn't introduce any cancer-associated mutations. The only variants that we see are ones that occur and are benign. They occur all the time in our own bodies, and they're benign. This has been confirmed a couple of times by other researchers now. The second stage is characterizing the iPS cells to make sure that they are pluripotent, induced pluripotent stem cells. The way that people used to do this about 7 years ago or so was to take cells, around 1 million cells, and inject them into an immunocompromised mouse. This is called the teratoma assay, and it relied on those cells around 2 months later forming a tumor, which you could then take out and analyze. When you do histological analysis, you can see that cells from all 3 germ layers have developed. So there are typical things like cartilage, nerve cells, and mesodermal derivatives, like muscle. We decided in the 2009 or so that there was probably a better way of doing this, so we developed the PluriTest diagnostic test for pluripotency. This was developed by my colleague Franz Joseph Mueller and another member of my group. This allows you to prove that the cells are pluripotent without using the teratome assay and not using animals, an animal-free assay. The way this works is that we've developed a website called pluritest.org. This website currently has 800 registered user sites. That is more than one investigator. Each institution is registered. We've had 20,000 data set uploads. These are gene expression profiles. It is now used in 29 countries, and in fact, we have licensed Thermo Fisher to do PluriTest with their microarrays last year. So our main objective was to make this so easy that people would not need any bioinformatics expertise. So it's an algorithm which works on the computer. It was based on machine learning, so it is quite reliable. The steps are to run a gene expression by RNA sequencing or an array. You upload the raw data to the PluriTest website, and then what comes out is a data analysis, including a diagram that resembles the one on the right. If the cells show up in that upper left-hand corner, that means that they are typical of pluripotent cells and therefore likely to be pluripotent themselves. As they show up anywhere else on this graph, they're probably differentiated or abnormal in some other way. We have developed an RNAseq version of PluriTest. Our first assay was based on Illumina's microarrays, but now we've adapted it to RNAseq, and we published the first use of that in a paper in a collaboration with one of our colleagues here in San Diego, to analyze 222 iPS cell lines using RNA sequencing. And you can see the picture from a figure from the paper on the right, which shows that most of the cell lines, but not all of them, gather in that upper left-hand quadrant. They're slightly off the main, the dark region in the red, because RNA-seq data give you slightly different results than microarray data, which is not surprising, but they are in fact pluripotent by this assay. On the lower left, you can see a little snapshot of the website, and over on the lower right, you can see the increase in RNA-seq, that is FASTQ files that have been uploaded to pluritest.org up to last month. That is now around 1,600. It's also been recognized by an international group called International Stem Cell Initiative, IPSC. We published this work this year as a collaborative effort to establish, to assess all these established techniques to determine the developmental and malignant potential of human pluripotent stem cells. And you'll see that this is a diagram from a figure in that paper showing that a new potential pluripotent stem cell line can go through first PluriTest and then followed by other assays to find out more about the cells. But the recommended test for initial characterization is, in fact, PluriTest. Okay, here's the issue I told you earlier. This is where a lot of things can go wrong during the expansion of the cells. And we started noticing this quite a few years ago, as did others in this field. We found out that copy numbers of genes would change during cell proliferation of human pluripotent stem cells. This just shows you one example from that paper. And we use SNP genotyping, which only takes a global view of the entire genome at pretty high resolution, higher resolution than karyotyping, but lower resolution than sequencing. And you can see just at a glance that in all the samples we looked at, there are many, many duplications of certain chromosomes, including chromosome 12, chromosome 17, and the X chromosome, as well as some others. So this is what has been shown to occur in many, many cell lines now all over the world. And so I think that is the incentive to come up with methods for checking the genomic integrity of the cells. This is one example about how easy it is to do this with SNP genotyping. This is a comparison of two different passage numbers. This is also in that paper of an embryonic stem cell line from the University of Chicago. You can see at the early passage at the top of that graph, all those little dots running down the middle mean that there's only one copy of the gene. If you look at later passage, you can see that single line has changed into two lines. That means there's an extra copy of this particular region of the chromosomes. This is a common duplication that many people have now discovered on chromosome 20. It occurs in pluripotent stem cells very frequently. Okay, so we've also shown that there are the mutations accumulate with expansion. This is a paper we published several years ago. And we first discovered that one of those mutations was a deletion of P53. If you culture the cells long enough, it's virtually inevitable that they will acquire P53 mutations. And the reason for that is that P53 gives the cells an advantage. They are more resistant to dying in the culture, and they probably divide a bit quicker. But if you just look at the data, every time a cell divides, about 5 to 7 changes occur in the genome. So as I said earlier, changes in the genome are perfectly normal, and we are, in fact, mosaics. We have lots of different genomes in our bodies. Most of those changes are benign because the genome is a really big place, and if you just randomly mutate it, you'll very seldom hit something that is dangerous. But some of the changes drive cancer, and it's especially important that there is accumulation of this mutation or loss of P53, because P53 is a gene that is mutated in about 50% of cancers. So essentially, if you lose P53, you lose the signal to tell the cells to stop dividing. And it also makes the cells, as I said earlier, resistant to stress. So P53 mutations provide a growth advantage in pluripotent stem cells, and therefore the frequency of mutations in pluripotent stem cells increases with cell expansion. The figure at the bottom is from a paper from Kevin Egan's lab, in which he obtained embryonic stem cell, banked embryonic stem cells from several different sources, ESI, Children's Hospital Boston, and YCell, and also the Sheffield group. And he found that in many cases, obviously not all, when these cells were expanded, when the passage number increased from left to right, the proportion of cells that had mutations in P53 increased. And you can see for the WA26 there, the increase appears to be exponential at the end, which is what you'd expect if these cells are taking over. They also found mutations in the WA9, also known as H9 cell line, which is very popular. H9 was the cell line that we used to show P53 mutations in the first place, and also used it widely for our SNP genotyping assays. And it also showed P53 mutations in this bank called MasterChef, which is from Sheffield University. Okay, the last stage, differentiation of the cells. Of course, the assays will be cell type specific. In our case, is differentiation to dopamine neurons. As I said earlier, we have developed really robust methods so that we can make dopamine neurons every single time from every single patient. These are little pictures of immunocytochemistry from 5 different patients showing Th positive and FOXA2 positive dopamine neurons. What we thought, again, that one shouldn't have to test for dopamine neuron function if you have an assay that will predict functionality of cells or cultures of cells. So early on, we obtained a group of samples from some of our collaborators in the G-Force labs, from Jun Takahashi, Lorenz Studer, Malin Parmar, and of course we have our own cell lines. And this is a principal component analysis that we did early on using microarrays, showing that the green cells, which are dopamine neurons at two different stages, day 13 and day 25, the dopamine neurons that you can see on the left from day 25 are yellow dots cluster with Jun Takahashi's red dot and the Studer's green dot. The earlier stage dopamine neuron precursor cells from our lab, day 13 to day 16, the yellow dots there, cluster with Malin Parmar's cells. Those are, she uses an earlier stage for transplantation, but it looks like they're on the same time course as our cells. And also in this diagram, you can see the neural stem cells, which are used by International Stem Cell in a trophic strategy for trying to rescue the remaining dopamine neurons in patients. Another kind of neuron, cortical neurons, undifferentiated iPS cells, and also the fetal mesencephalon, which was the tissue that was used to treat Parkinson's patients in the 1980s and 90s. So now we're in the process of developing a PluriTest-like algorithm, which we call Neuro Test. Again, it's gene expression-based and quality control assay. Like PluriTest, the cells are validated by non-genomic criteria. We use flow, neurotransmitter release, and animal studies. Like PluriTest, Neuro Test is intended to replace the animal and the in vitro assays of cell line equivalents. And if you see, there's a heat map on the left, which just shows that we designate cells as passing or failing other assays, and then we do gene expression on them. And you'll see that we have turned the PluriTest algorithm on its head so that the dopamine neurons will turn up in the upper left-hand quadrant like the pluripotent cells did in PluriTest. In the middle, at the very bottom right-hand corner, are the iPS cells they came from, and in the middle, there are different stages of differentiating dopamine neurons on their way to becoming day 25 transplantable cells. I just wanted to show you that this is one of the ways that we find out whether the cells are functional or not. This is the hardest way. This involves taking dopamine out of an animal, a rat in this case. This is an immune-suppressed rat, and we've killed off all the dopamine neurons at one side. That's the upper left-hand corner. The upper right-hand corner shows our transplants of human cells, and it's important to notice that those dark staining cells there are the human dopamine neurons, and that they stay right where we put them, which is very important. The animals recover from the loss of dopamine, so those cells can compensate for the loss of dopamine on that side of the brain. And if you look up very closely, you can see that the cells that are in this very tight configuration actually are sending axons out throughout the striatum. As I mentioned earlier, I just want to bring this back full circle and remind you that using autologous therapy allows for simple cell manufacturing. One 6-well plate will give us 50 to 60 million dopamine neurons. That's plenty for our transplantation of 2 to 5 million cells and all of the QC that we need to do. In contrast, for heart restoration plans in progress, they are requiring 1 billion cardiac cells. International Stem Cell Inc., which is using a neurotrophic approach to try to rescue the remaining dopamine neurons, plans to transplant 70 million neural stem cells into patients' brains. So this allows us, this lack of this simple cell manufacturing allows us to scale out rather than up. We don't need to produce millions of cells, and so we're testing in a collaboration with Scaled Biolabs in the Bay Area in a project that is funded by CIRM, California Institute for Regenerative Medicine. We are testing reagents in parallel using very small input, low number of cells, and low reagent cost. This allows us to optimize the differentiation conditions without spending a ton of money. With that, I want to acknowledge the people in my lab. This project in particular, Andres Brat-Leal is directing it. Roy Williams is in charge of the bioinformatics, and the rest of the people in the lab all collaborate. I also have outside collaborators that are listed on the right. And I just want to close by showing my lab with another one of our collaborators. We are at the Wild Animal Park meeting with our collaborators to try to rescue the northern white rhino. One of our collaborators is that rhino, which you can see behind us. She's a southern white rhino. She's been brought in to serve as a surrogate if we can produce embryos from iPS cells that we've made into gametes. So thank you for your time. I am finished.